Within the structure of a cell membrane, the phospholipid heads face blank, while the tails face blank. Okay. Well, the tails face the inside of the cell. So the tails, hold on. Let me do. So let me back in the picture. Within So the phospholipid heads face each other while the tails face the interior of the membrane. So let me get the question here. This is a different question. Let me see. So that's cytosol, extracellular. So it's gonna be the phospholipid heads place the cytosol because they face inward, and the tails face each other. The tails face each other. The prefix, okay, you know this one. Which one means cell? Yes. Okay. Which of the following is the meaning of the prefix endo? Endo.
Maybe within.
Yes. Within. Right. Nice. Okay. The prefix blank means above, as well as greater than or higher amounts in comparison between solutions. Which one means greater than, higher than, or more than? You see it? Which one means above?
Hyper.
Hyper. So hyper is the answer there. Yeah. Good job. Nice. The prefix for below is
Would it be hypo?
hypo. Very good. Nice. The smallest unit of organisms like humans that is considered to be alive is
Cell.
cell. Very good. Nice. In comparing two solutions of the same volume, the one with more solutes has less or more water than the one with fewer. If it has more solutes, it has more stuff in it, so it has less water than the one with fewer. It has less water in there. Good. Okay. What is the cell part? Okay, what is it pointing at? The black arrow.
The nucleus.
That's right. It's pointing at the nucleus. Good job. Nice. Okay. The cell membrane. Okay, so there might be more than one answer. The cell membrane can perform functions related to DNA replication, detecting signals, attaching to other cells, or controlling the movement of substances entering the cell. You're in recharge right now. Did you mean to be there?
Um,
You're
um,
not doing the exit recharge. You're doing the questions over again.
oh, I thought it was the assignment from yesterday.
It was, but you had it in recharge, um not the actual, you know what I mean?
Mhm.
You were doing recharge for chapter 3, I think. Okay. That's okay. It's good for us to practice. Okay. What term by definition means the ability to work, to do work? Reaction, equilibrium, energy, or respiration? I think energy is the ability to do work. When you have energy, you can work. When you don't have energy, you can't do work. Okay. Enzymes are able to interact only with a particular type of molecule called its what? What is that? Remember the lock and key idea? What is that piece that the enzyme attaches to? Do you remember what it's called? If not, you can look it up. Okay, so here, each type of enzyme is specific, acting only on a particular type of molecule called its substrate. Substrate. So that's the word that they want. Good. All right. Now we're up to the chemical energy of molecules is held in the blank between the atoms. Okay, so chemical energy is held in the bonds. There's that word, bonds, between the atoms of the molecules. So the word there is bonds. Yeah. Good. A series of chemical reactions called blank pathways are enzyme catalyzed reactions that occur in a specific sequence. Blank pathways are enzyme catalyzed reactions that occur in a specific sequence. Okay, so here it is. Specific enzymes catalyze each of the hundreds of different chemical reactions that constitute cellular metabolism. Sequences of enzyme-controlled reactions called metabolic, you want to put the first word, pathways was already given to you, called metabolic pathways lead to synthesis or breakdown of particular biochemicals. So metabolic. Good. The high energy storage of ATP exists in the bonds between what kind of molecules? Each ATP molecule includes a chain of three linked chemical groups called phosphates. As energy is released during cellular respiration, some of it is captured in the bond that attaches to the terminal phosphates to the end of the rest. Terminal phosphate to the rest of the ATP molecule. This attachment is described as a high energy bond. When energy is released for a metabolic reaction, the terminal phosphate bond breaks, releasing the stored energy. The second phosphate bond is high energy too. So it's phosphate. Phosphate. Oh wait, it says molecules. So between the, I'm not sure if that's what they want, but it's phosphate bonds. Let me see. Let me see if that's the word they want though. Because Google search this. Hold on, what's the question? Exists in bonds between the blank. Okay. Um, exists I'm not sure if they want. Yeah, just put phosphate and see what happens. Put phosphate, because I'm not sure what they want. P, yeah, there you go. Try that. Because then, is that what they wanted? Okay, because it's the bonds, and I didn't know if they wanted phosphate bonds or just phosphate. Okay, that works. Okay, which of these three factors? Okay, this was a question from yesterday. They're giving it back to you. Which of the three of these factors can denature an enzyme? Yeah. Streams in pH. What else? Two more. Heat, right? And what else? No. Radiation is the other one. Radiation, extreme pH, and heat are the three for that. Like most proteins, okay, they want the word denatured here. So make sure you get the form correct. Denatured. ED. Uh-huh. Yeah. Should take it this time. There you go. Nice. All right. List the three end products of cellular. Okay, we haven't had this question before. The three end products of cellular respiration. Okay. Cellular respiration consists of three distinct yet interconnected series of reactions: glycolysis, the citric acid cycle, and the electron transport chain. Glycolysis and the electron transport chain are pathways similar to the general pathway shown in figure 4.6. Figure 4.9 depicts how a metabolic pathway can form a cycle, such as the citric acid cycle, if the product reacts to reform the original substrate. Glucose and oxygen are required for cellular respiration. The following is the overall summary reaction. So if you look at cellular respiration, it's given to you glucose, which is C6H12O6, plus oxygen. So you put in glucose and oxygen, and you get carbon dioxide, water, and energy, or ATPs. Okay, carbon dioxide, water, and energy. I think it asks for the product, right? Let's look at it. So yeah, the three end products are carbon dioxide, water, and energy, based on the formula that we saw. And energies. Good job. By definition, blank is the capacity to change something. It is the ability to do work. Okay, so now it's fill in the blank. What is that word for the ability to do work?
Or would it be energy?
Energy, right. Energy is the capacity to change something. Very good. The products, okay, here they're asking it in a different way. The products of cellular respiration include H2O, ATP, and what? look at the formula, look at the right-hand side of the formula, and fill in the box that's missing. So on the right-hand side, there are three things, right? The question gave you two of these things. Which one did you not see on that list?
There would be energy.
Energy. It's another way of asking for, because water is H2O. ATP is energy. Actually, ATP is energy. So they gave you that. The other one is carbon dioxide or CO2. I don't know if they want. Probably it doesn't matter if you give them the word or if they give you the word or the formula. That's correct. Yeah, they took it. They would have taken, yeah, see, they would have taken CO2 as well. Remember that energy and ATPs are the same thing. Okay, for this context. Okay, next thing. Where is chemical energy held within a molecule? Within the nucleus, at the site of the electrons, or within chemical bonds? Chemical energy is held in the bonds. I'm reading under release of chemical energy, the first sentence. Chemical energy is held in the bonds between the atoms of molecules. Okay. Right. The DNA in cells holds information most directly related to the blank. Sequence of proteins, formation of cell membrane, or double helix structure. Okay. Enzymes control essential metabolic reactions. Therefore, cells must have instructions for producing enzymes as well as other types of protein. The sequence of building blocks of deoxyribonucleic acid, DNA molecules, hold the information to manufacture proteins in the form of a genetic code. DNA molecules pass from parents to offspring when a sperm fertilizes an egg. As the offspring grows and develops, mitosis passes the information in the DNA sequence of the chromosomes to new cells. So DNA molecules hold the information to manufacture proteins. Is what it said. Directly related to the sequence of proteins. I would say it's the sequence of proteins. Based on what I read there. Yep. Okay, so what molecule does this look like? what molecules is the picture of? Adenosine triphosphate, triglyceride, deoxyribonucleic acid, or cholesterol? What is that a picture of? You look at figure 4.7, it says ATP at the bottom, which is adenosine triphosphate. Okay. Next question. What is the name of a segment of DNA that codes for a particular peptide, polypeptide? What is the name of a segment of DNA that codes for a particular polypeptide? Gene, genome, chromosome, or nucleotide? Okay, so a complete set of genetic instructions is the genome. All cells except the sex cells contain two copies of the genome, one inherited from each parent. Segments of the genome that encode proteins are called genes. Only about 1.5% of the human genome encodes protein and is called the exome. Much of the rest of the genome controls when and where genes become active to provide protein synthesis. Clinical application 4.1 describes how exome sequencing aids disease. Okay, keep going, because I'm not sure that that's what they wanted. Go down. Recall from section 2.6 that the building blocks of nucleic acids are nucleotides. They are joined so that the sugars and the phosphates alternate, forming a long backbone to the polypeptide chain. DNA has two such polynucleotide chains. In a DNA molecule, the nitrogen bases bond weakly to the bases of the other strand. The resulting structures like a ladder where the uprights represent alternating sugar and phosphate backbones of the two strands, and the rungs represent the bases. In a nucleotide, the DNA nitrogenous base may be one of four types: adenine, thymine, cytosine, or guanine. A gene is a sequence of nucleotide bases along one DNA strand that specifies a particular protein's amino acid sequence. Okay, let's look at the question. And let's see what it says. I would say that's a gene, a segment of DNA that codes for a particular polypeptide is a gene, based on what I read there. Radiation, okay. Radiation, heat, and some chemicals can cause a protein to unfold, called denaturation. So it's the form of the word that they want. Uh-huh. Good. Okay. The building blocks of nucleic acids are blank, made from a phosphate and a sugar. Or what? The building blocks of nucleic acid. Okay. So, okay, so the building blocks of nucleic acids are nucleotides. So it's this second word here, nucleotides. They are joined so that the sugars and phosphate alternate. Uh-huh. Water, carbon dioxide, and energy are the end products of the process of, what did we read about where those were the end products?
Would it be protein synthesis?
It's cellular respiration. You can look at it again and you can see it. So here, see, whoops, see? Cellular respiration consists of 3 interconnected processes. So here's your process, okay? The following, if you look at the bottom sentence, the following is the overall summary reaction of cellular respiration. Glucose plus oxygen gives me carbon dioxide, water, and ATP. So it's cellular respiration. Okay, cool. The 2 components of the backbone of DNA are phosphates and
Sugars?
sugars. Very good. Yay! All right, cool. Moving along. Okay, what is the right formula? Okay, so we have glucose plus oxygen yield carbon dioxide, water, and energy. So glucose plus oxygen, yeah. Glucose plus, okay, wait a minute. You need glucose plus oxygen plus energy. It's the bottom one. Glucose plus oxygen plus energy, because the reason it's not the third one is because you only have glucose. Wait a second. I'm getting confused now. Because there should be 3 products on the right. Glucose plus oxygen. Okay, so we know it's not the first 2 because we need glucose plus oxygen and we don't have that. So glucose plus oxygen. Yeah, you were right. It's the 3rd one. I was wrong. It's the 3rd one. You were correct the first time. I had to stare at it because I didn't see the arrow right away. But yeah, that's right now. There you go. It's balanced. Okay. All right. How many nucleotide chains are found in a DNA molecule? How many nucleotide chains are in a DNA molecule? Okay. Recall for, okay, building blocks. The building blocks of nucleic acids are nucleotides. They are joined so sugars and phosphates alternate, forming a long backbone. DNA has 2 such polynucleotide chains. Two nucleotide chains. The genetic code is found in molecules of RNA. Okay. Okay, keep going. I think it's DNA. So I would say it's false because that should be DNA, not RNA. Right. The DNA molecule is 2 strands that connect via rungs of nucleotides as a ladder. The ladder includes okay. So where's my ladder? So here, the DNA molecule, whoops, I need to read so in a DNA molecule, the nitrogenous bases, so we have nitrogen bases, right? Bind weakly to the bases of the other strand. The resulting structure is like a ladder, where the uprights represent the alternating sugar and phosphate backbones of the two strands, and the rungs represent the nitrogenous bases. Okay. Okay, so let's go back. Okay, so it contains sugar and phosphate and amino acid. Oh, they don't want to connect the amino acid. Okay, so they just wanted what was on the rungs, I guess. Sugar and phosphate that connect. The amino acids are the rungs, but they were just talking about the two strands that connect it. Okay, that was my fault. Not all DNA molecules encode proteins, but the segments, okay, I don't remember the specific word for the DNA that can encode, it's like an exosome or something like that. Yeah, okay. Only about 1.5% of the human genome encodes protein, and it is called the exome. Exome. E-X-O-M-E. Exome. Exomes. I would make it plural. Yeah. See if that's, no? Oh, they wanted that. Okay. I thought, hmm. I thought they told us, okay, we have to read about it first. They're telling us we have to read it. So we have to open it and reread it, even though that's what it said. Before they let us go on, we have to read it. They wanted genes for that question, but we have to open it up and read it, even if we don't read it. Yeah, okay, they highlighted it for us here. But I took the word below it. Segments of the genome that include proteins are genes. Okay, that. So it was looking for that piece. They highlighted it for you. So the segments of the genome are genes, and then okay, and it is called the exome. Okay, I don't know why. I guess the exome is the whole, the exome is a specific part. I'm not sure here. Segments of the genome that encode proteins are genes. And only about 1.5% of the genome encodes protein. Okay, I don't know what that difference is, but okay. I'll have to look at it. But genes is okay, so we can go on and then we'll have to come back. Nucleotides are the building blocks of what type of organic compound? DNA and RNA. Yeah, that's what we're reading here. Okay, now, this is something that we kind of read, but not really. What nitrogenous bases are found in DNA? Adenine, cytosine, guanine, and thymine. Cytosine, uracil, adenine, and thymine. Thymine, guanine, uracil, and adenine. It's the first one. Adenine, cytosine, guanine, and thymine. Okay. Okay, the backbone of the DNA molecule is found by alternating phosphate and what? What's the other rung made out of? Okay, so that the sugars and the phosphates alternate. So the other word would be sugars. Sugars is the other one. The type of nucleic acid that is in the form of a double helix is
Would it be DNA?
DNA, right? Good. Nice. What type of nucleic acid is double-stranded? DNA, right? Because RNA is a single strand. It's one piece. DNA is two pieces inclined together. Good job. The two strands of a DNA molecule are bonded together at what locations? Between nitrogenous bases, between deoxyribose sugar groups, or between phosphate groups. So where do they come together? Where do they connect together? So, in a DNA molecule, the nitrogenous bases project from the backbone and bind weakly to the bases of the other strand. The resulting structure is like a ladder where the uprights represent the alternating sugar and phosphate of the two strands, and the rungs represent the nitrogenous bases. Okay, so scroll down below that. I want to see what the pictures says. The caption says. DNA structure, the molecular ladder of its run twists into a double helix. The ladder's rungs consist of complementary base pairs held together by hydrogen bonds. A with T or T with A and G with C. Okay, let me see the question again so I can see what they asked us. Okay, hold on. Because I'm not sure what they. Let me look this. Um, the two and the two strands hydrogen bonds between pairs of bases. So that's what I'm saying. But I would say between nitrogenous bases is your best bet. Held together between nitrogenous bases. Because they're not sugar groups and they're not phosphates. So the bases are the rungs that are the rungs of the ladder. A DNA molecule, okay, that's gonna be nitrogenous bases. That's a different way of asking us the same question that they just asked. A DNA molecule has a structure that looks like a trusted ladder. What forms the rungs of this ladder? Okay. Next question. What is the name of a segment of, okay, that's, let's not get this wrong again. It codes for a particular polypeptide. That's a gene. The name of a segment of DNA that codes for a particular polypeptide is a gene. Okay, now we're gonna match the bases. So adenine always pairs with guanine, the G. A pairs with G and C pairs with T in DNA. You're going to see that in RNA, one of these is going to change, but in DNA, A always pairs with G And C always pairs with T. So. Oh. Adenine, I got it wrong? Pairs of DNA. Adenine with guanine and okay, let's review it because I don't remember I thought that's what I remembered seeing. I got it wrong. I'm sorry. These combinations, A with T and A with T and G with C. I read it wrong because I thought T and G together. Okay. A with T, like at. A with T and G with C are called complementary base pairs. Okay. All right, we can fix that later. RNA is different. A with T and G with C. Okay, let's go to the question. They'll give it back to us. So we can do it in a little bit. List the nitrogenous bases found in DNA. Okay, so there's 4 of them. Do you remember what they are?
Adenine,
Adenine,
guanine,
guanine, right? 2 more.
uracil,
Not in DNA. That's in RNA. Thymine is the one, and cytosine. Uracil is in RNA. A, T, and G with C, guanine and cytosine. Okay, so let's do that. Okay. Prior to cell division, the genetic information contained in blank must be replicated. Prior to cell division, the genetic information contained in blank must be replicated. We can review it. When a cell divides, each newly formed cell must have a copy of the original cell's genetic information, so it will be able to synthesize the proteins to build cellular parts and metabolize. The copying of DNA is called replication, and it takes place during interphase of the cell cycle. When a cell divides, each newly formed cell must have a copy of the original cell's DNA. So let's see. Prior to cell division, I would say DNA must be replicated. You have to replicate it first before you divide it. You have to copy it before you split it. DNA has a molecular ladder chain or ladder structure known as the blank. 2 words here. Okay, so let's go ahead and review. So the resulting structure is like a ladder, and the rungs represent the nitrogenous bases. Okay. Recall from that the building blocks of nucleic acids are nucleotides. They are joined so that the sugars alternate, forming a long backbone to the polynucleotide chain. I think they're looking for polynucleotide chain. Forming a long backbone to the polynucleotide chain. DNA has two such polynucleotide chains. I think they're looking for that. So it would be polynucleotide. NU, it's misspelled. NUCLE. OTIDE. And then the second word is chain. There you go. Let's see if that's what they wanted. Is it? No. Oh, double helix. Okay. All right. So you wanted the double helix structure. That's true too, but yeah, that could have been. All right. That's just a little bit picky on their part. Because the polynucleotide chain forms. Okay, now we know what they want. But they didn't make us read about it again. So let's go. We can come back. The double strands are linked by chemical bonds formed between the blank. Okay, so let's review it. Chemical bonds formed between the what? Chemical bonds formed between the okay, I'm not sure if they want, I think they want sugars and phosphates. Let's take a look. See if that's what they want. Let's see if that's what they want. Go to the question. Either that or it's nitrogenous bases. Okay, they don't have and the, so maybe it's maybe they want nitrogenous bases. They're forming, they're bonds between the sugars and the phosphates, but I think they're talking about nitrogenous bases here because they don't have a third word. They have only two. So I'm not sure if that's because it said nitrogenous bases in the let's try that. Let's see if that's what they want. Yeah, okay. Nitrogenous or complementary. Okay. That makes sense. Replication is the movement of DNA to new cells during mitosis. Duplication of DNA during interphase or the copying of RNA from DNA. Replication is the duplication of DNA during interphase. Replication means to duplicate it. You're duplicating the DNA. The copying of RNA is something different. That's part of it too, but replication is the duplication of DNA. Yeah, okay. The DNA molecule is two strands that connect via rungs of nucleotides in a ladder. The ladder includes, okay, they want sugar and phosphate for this. The first step of DNA replication is, okay, I don't know if we got to this step yet. We'll see. When you replicate DNA, when you're going to make a copy, what do you do first? Okay, as replication begins, I'm reading in the middle of the page. As replication begins, the hydrogen bonds between complementary base pairs in each DNA molecule break. So you break the bonds between the base pairs, then you unwind the helix, and then you pull the strands apart. So you break hydrogen bonds between base pairs, you unwind the double helix, and then the bases fall, the bases are exposed. So you break the hydrogen bonds between bases first before you unwind the strand. Okay, whoops. But you just told me they break first before they unwind. This is confusing. It's confusing me. So if it's confusing you, I don't blame you. Because it just said they break the hydrogen bond between, but I guess it's the, go back to the reading because I don't know if yeah, I read that. The hydrogen bonds between complementary base pairs break. The double helix unwinds. Then, yeah, so hydrogen bonds between complementary base pairs in each molecule break. That's what it said. And that's what I put. And you said it was wrong. I don't know what to tell you there. I don't know what to tell you. Yeah, I don't know what to say about that. You have to break the bonds before the helix can unwind. I'm not sure, but if we get the question again, we'll know what we should put, unless they reframe it for you. Okay, let's go back. When A binds with T and G with C, this pairing is termed what? When A binds with T and G binds with C. Let's look up the word. Review the resource because I don't know what word they're looking for. So let's read the book and see what it said. Let's review what it says. When A binds with T and G binds with C, okay. Okay, in a DNA molecule. Okay, did they tell us what it's called? In a DNA molecule, the resulting. Okay, keep going. The base pair. Well, let's see. These combinations are complementary base pair. Let's see what the question says. This pairing is termed when A binds with T. I don't know if they're looking for okay. When A binds with T and G binds with C and DNA. The pairing is termed I would say complementary is what they're looking for, probably. Complementary. That makes the most sense. With what? Okay. Before cells can divide, the blank must replicate so that each cell will have a complete genome. Look at your resources. We read this earlier. I think this is DNA. DNA replicates so that each cell will have a complete genome. Okay. What is produced by the process of DNA molecules? Okay, so we have to read this and see what the product of replication is. So, okay. Okay. In this way, a new strand of complementary. Okay, so the double-stranded structure of DNA makes replication possible. As replication begins, hydrogen bonds between complementary base pairs in each DNA molecule break. The double helix unwinds and the two strands pull apart, exposing the nitrogenous bases. Then, an enzyme called DNA polymerase catalyzes the joining of new DNA nucleotides, which form complementary pairs with the exposed bases on the separated strand. Other proteins and enzymes are involved in various ways to assist this complex replication process. And in this way, a new strand of complementary nucleotides forms along each of the old strands. So we have two old strands, and we have a new strand of complementary nucleotides on each. Okay, keep going. Does it say anything else? Okay, so does it say anything else on this page? Okay, that's our picture. DNA forms two double helices from one. When a DNA molecule replicates, its original strands separate locally. A new strand of complementary nucleotides forms along each original strand using free nucleotides in the cell. DNA replication, okay, this is the question here. DNA replication produces two complete DNA molecules, each with one old strand of the original molecule and one new strand. During mitosis, the two DNA molecules that form the two chromatids of each of the chromosomes separate. So we have one old strand and one new strand, is what it says here. So let's go to the question. Two complete DNA molecules, each with one old strand and one new strand. So the first part, the first option is what we read. Okay, next question. The type of nucleic acid that is in the form of a double helix that's DNA, deoxyribonucleic acid. Sometimes we get a little redundant. Okay, next question. Okay, now this is a different process. This is transcription. Transcription produces a molecule of blank from a template of DNA. So let's look at that. DNA molecules stay in the nucleus of a cell, maintaining the genetic information. However, protein synthesis occurs in the cytoplasm. The genetic information reaches the cytoplasm by being copied into molecules of RNA. RNA connects at the nucleus because the molecules are much shorter than the DNA that composes entire chromosomes. The process of synthesizing RNA is called transcription. Okay, so you make a copy of RNA from DNA. That's what it says, basically. So RNA is the answer to that question. Oops. RNA. Okay, oops. Oh, they wanted the actual, okay, so RNA is correct, but they wanted it more specific. They wanted mRNA, so messenger RNA. RNA is not wrong. They just wanted a more specific answer than mRNA is messenger RNA. Because there's different kinds. So they wanted messenger RNA for that. Okay, let's continue. What is DNA replication? So, okay, replication is copying DNA, process of copying DNA. That's what replication is. It doesn't have anything to do with making proteins. That's a different, because that's not what replication does. Okay, so okay, next question. The term blank refers to the process of synthesizing messenger RNA. That's what the M means. The term blank refers to the process of synthesizing messenger RNA within the nucleus of a cell. Let's look at the book. Okay, so scroll down a little bit because we haven't seen okay, so the process of synthesizing DNA molecules, okay, execute, okay. In addition, the RNA that connected is single-stranded. Okay. The process of okay, what was the question again? Go back to the question itself. The term blank refers to synthesizing mRNA. Hold on. Yeah, that's transcription. Transcription. So they're just giving you another way of another way of asking it. That's transcription. Okay. Next question. Drag and place in order the events that occurred during. Okay, so okay, so the the steps in DNA replication. Let me find the steps. Okay. Okay. So let me see. Let me see if it tells me something here. DNA replication. Let's see. Okay, so the double helix unwinds. That's first. So that would be yeah, the DNA, and then the hydrogen bonds break, and then okay, so the DNA strands unwind, the hydrogen bonds break after that, and then the nuclear, let's see, the complementary nuclei. I would say the third one would be the last one on the list. So flip the bottom two. The DNA strands, hydrogen bonds break because they have to, and then the complementary nucleotides. I don't know if the nucleotides pair up before or after they break. Let me look. Hold on. Let me see. The complementary nucleotides pair up with exposed bases. So they unzip, then the see. Follow. Let me see if I can actually follow this. Before replication can start, the double strands of DNA need to separate from each other. An enzyme called DNA helicase breaks the hydrogen bonds that connect that. Okay. I'm not sure if it's I'm not sure which one it is. Um, so let's see. Oh, it did work. Okay. The DNA strands unwind, the hydrogen bonds break, complementary nucleotides pair up. Okay. In DNA, adenine is the complementary base to, okay, so that's gonna be thymine. Thymine. First box is thymine. And the guanine is the complementary base to cytosine. No, cytosine. You have guanine already. Uh-huh. Cytosine. I make sure it's spelled correctly. I think thymine is misspelled, and I think cytosine. They're the right words, but I think they're misspelled. Look at the spelling, because I think it might be. Thymine is thymine. 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 And cytosine is Cytosine. Cytosine. There you go. The molecules which transcribe the molecule which transcribes the genes of DNA and moves them from the nucleus, specifically, okay, they're looking for, I think let's look it up, but I think they're looking for mRNA here. Let's look up the passage in the book, but I think this is the one where they're looking for mRNA. mRNA is the type. Yeah, so the word in blue parenthesis, small m with RNA next to it, I think is what they're looking for here. mRNA is the small m and then RNA. I think that's what they want right here. Okay. Okay, so the products of DNA replication are two DNA molecules, each with one old strand and one new strand. You get two DNA molecules with one old strand and one new strand. Okay, you can look at it. So it says, in this way, a new strand of complementary nucleotides forms along each of the old strands. So you have two old strands that are now apart, and each of them gets a new strand, like a new side of their zipper. So they get one old one and one new one. If you scroll down, you'll see it. Scroll down. Scroll down. Down. Okay, look. DNA, no, below where it says watch video, look. DNA replication produces two complete DNA molecules, each with one old strand of the original molecule and one new strand. One old strand and one new strand. Okay. Yep. Okay. An example of a single-stranded nucleic acid. So which one is single-stranded?
DNA.
Single-stranded is RNA.
Oh.
Double-stranded is DNA. D for double, right? RNA is single-stranded. We haven't talked much about RNA yet. I'm sure we'll get to it soon. Yeah. Okay, so transcription is defined as, okay, so transcription is your your transcription, okay, transcription and translation. We've talked about replication, but now we're going to talk about transcription and translation. So we need to review because we haven't talked about this yet. Okay. I need to read it so I can tell you. So, transcription is DNA molecules stay in the nucleus of a cell, maintaining the genetic information. However, protein synthesis occurs in the cytoplasm. The genetic information reaches the cytoplasm by being carried into molecules of RNA. RNA connects with the nucleus because the molecules are much shorter than the DNA that comprises entire chromosomes. In addition, the RNA that carries a gene's information is single-stranded. The process of synthesizing, putting together RNA is called transcription. Messenger RNA is the type of RNA that carries a gene's message out of the nucleus. RNA molecules differ from DNA. Okay, so synthesizing RNA is transcription. Let's scroll down. Okay, so synthesizing RNA is transcription. Okay, so let's go to the question and see what we can see here. Transcription is defined as synthesizing RNA from DNA. It's the second one. You synthesize RNA in transcription, and then you use RNA to make proteins in translation. Okay. Okay, they just asked you this. Transcription is the process of making RNA from DNA. That's RNA. Okay. Name the nitrogenous base found in DNA. Okay, so what's the one that we don't find in DNA?
I think it's uracil.
That's correct. Yeah, that was an easy question. Okay, during DNA replication, complementary blank pair up with the nitrogenous bases that are exposed as the DNA helix unwinds. I was going to say complementary bases, but they have complementary blank pair up with the nitrogenous bases. Let's review it. I'm not sure what they want. Complementary what? Hydrogen bonds between complementary base pairs break. Okay, so thus, okay, so hydrogen bonds between complementary base pairs break. The double helix unwinds and the two strands pull apart, exposing the nitrogenous bases. Then, an enzyme called DNA polymerase exposing the nitrogenous bases. Then an enzyme called DNA polymerase catalyzes the joining of new DNA molecules, which form complementary pairs. Complementary pairs. Let's see the question again. I don't know. Because I would have said, mm, mm. Yeah, I don't know what word I'm looking for. During DNA replication, complementary blank. Okay, so let me see. Um, during DNA replication, complementary blank. What is it? Pair up with the nitrogenous bases. Pair up with the nitrogenous bases. That are exposed. That are exposed as the DNA double helix unwind complementary blank. Let me see. It must replicate Hmm. I don't know. I don't know what word they want there. Yeah, I don't know. Try bases and see. I don't know what I don't know what they're looking for there. Okay, complementary nucleotides or okay, they wanted nitrogenous bases. Okay, again, like they wanted a more specific answer than we gave. So at least we know um nitrogenous bases. Okay. Okay. Go to the next. It's not wrong. It's just not as specific as they wanted it. So I don't know. What kind of RNA carries the genetic information from the nucleus to the cytoplasm? Okay, let's look at it because we have two kinds. We have actually we have three. There's ribosomal RNA, there's transfer RNA, and there's messenger RNA. So let's look at the concept resources. Look at the review. I think it's T, but I'm not sure. Okay, so messenger RNA carries a gene's message out of the nucleus. Keep going. And then here's the comparison of DNA to RNA. Okay, keep going. The enzymes, for example, okay, then this is the complementary base pairs RNA. Keep going. Each amino acid is specifically the bases are represented by a codon. The mRNA constitutes the genetic code. In addition to mRNA, okay, so hold on, let me do. Okay, so the type of RNA that contains information, it carries the information from the DNA to the cytoplasm. That's mRNA. mRNA takes it out of the nucleus into the cytoplasm. So that's mRNA. mRNA. All right. What determines the order of amino acids in a protein? Okay, let's look. I don't think we've seen this yet. The order of amino acids in a protein. Okay. Cells can synthesize specific proteins because the sequence of nucleotide bases in the DNA specifies a particular sequence of amino acid building blocks in a protein molecule. This correspondence of gene and protein building blocks is called the genetic code. Recall from section 2.6 that a biological protein may be built of 20 types of amino acids. Each amino acid is represented in a DNA molecule by a particular sequence of 3 nucleotides. The DNA sequence GGT represents one type of amino acid. GCA represents another, and TTA is a different one. Other nucleotide sequences encode the instructions for beginning or ending the synthesis of a protein molecule. Thus, the sequence of nucleotides in a DNA molecule denotes the order of amino acids in a protein molecule. So it's the sequence of nucleotides in a DNA molecule. The sequence of nucleotides in a DNA molecule denotes the order of amino acids in a protein. The sequence of nucleotides in a DNA molecule denotes the order of amino acids in a protein molecule, as well as where to start or stop synthesis. Okay, what does our question say? The sequence, okay, we didn't read far enough. Keep going. Let's keep reading it. And we'll see what we have here. DNA molecules stay in the nucleus of a cell. However, protein synthesis occurs in the cytoplasm. The genetic information reaches the cytoplasm by being copied into molecules of RNA. Okay. The process of synthesis. Okay. RNA molecule. Okay. Most RNA molecules are single-stranded. Each, however, whereas that in place of thymine. Okay. We read that too. And we read that. When RNA polymerase reaches, so the enzyme RNA polymerase synthesizes messenger RNA following the rules of complementary rearrangement. Specific DNA sequences outside genes signal which of the two RNA molecules, which. Okay, let's go back to the question again. Let me put it in and see. Okay, the sequence of nucleotides in the DNA encoding it. Okay, let me see the question here. The sequence. I would say it's the order of the bases within a gene. Because yeah, I think that's probably the best that I can think of. Because the bases are going to tell you what the complementary bases are. Try that. See if it's correct or not. Okay. Which type of, okay. Which type of nucleic acid is double-stranded and contains the sugar deoxyribose? Which one is double-stranded?
DNA.
Uh-huh. Yeah, they really want you to get that. Okay, next. The three base nucleotide sequence that corresponds to an amino acid. Okay, I don't know the answer to this question. The three base nucleotide sequence that corresponds to an amino acid. Cells can synthesize specific proteins because the sequence of nucleotide bases in the DNA of genes specifies a particular sequence of amino acid building blocks. This corresponding building block sequence is the genetic code. Okay, so DNA molecule, okay. And the three Okay, so it says each amino acid is represented in a DNA molecule by a particular sequence of 3 nucleotides. Let's see if that's on the list. Let's see. Or do they need us to be more specific than that? Oh, they need the word for that 3, but okay. The 3 base nucleotide sequence. The 3. Okay. Let's see. Nucleotide sequence. Oops. Sequence on RNA. It's called a codon. CODON. It's a codon. Okay. Next question. Using the genetic code DNA base sequence to build mRNA. Okay, so they're talking about the process. Using the genetic code DNA-based sequence sequence hold on. To build mRNA occurs. Okay. To build mRNA occurs during that's so that's transcription. Transcription is the first box. And producing a protein based on the order of an mRNA. Okay, so producing a protein from an mRNA occurs during Translation. Translation is where they make proteins. Yeah. There are blank possible codons and blank different amino acids. Therefore, more than one codon can code for a particular amino acid. Well, okay. I don't remember seeing this at all. But let's see. Do you want to stop here? You stopped screen sharing, so I
Um,
can go until 12 if you want, but it's up to you.
we could stop here.
Okay. All right then. We got pretty far, I think. You should be able to get through the rest of chapter 4 pretty easily. Has she lectured on 4 yet or not yet?
No, not yet. She was almost done with like chapter 3.
Oh, okay. Okay, so you're ahead of her. So I'll see you this week, later this week.
Yes.
All right, Daniela, take care of yourself, okay?
Okay, thank you.
Bye.
Bye.